Before jumping into the visual system, I want to do a brief overview of neuroanatomy. So, does anybody remember what the frontal lobe does? Yes, it does decision-making, planning, problem-solving, personality, and emotional regulation, as well as motor control. Okay, what about the parietal lobe? No guesses for the parietal lobe? Okay. The parietal lobe, it processes touch, pressure, pain, and temperature. It actually also has some relation to facial recognition and memory storage. And finally, the occipital lobe. So for the rest of the lecture, consider where each part happens throughout the lecture. So any visual input travels from the front of your head to the back of your head. Okay. So the electromagnetic spectrum is something we have to review. So it's the waves of energy traveling at the speed of light. For humans, we can see about 700 to 400 nanometers, and violet is shorter or more energy, and red is longer or less energy. So before our brains process visual information, a transfer of energy must take place. Phototransduction is the process of converting physical energy, in this case light waves, into neural signals that the brain can process. So again, this is the electromagnetic spectrum. We can only perceive a narrow slice of it, 380 to 750 nanometers, violet to red light. So wavelength is perceived color. Shorter wavelengths are violet or blue, which are high frequency, more energy. Longer wavelengths are red, low frequency, less energy. Amplitude is related to perceived brightness, so taller waves will be brighter light and shorter waves will be dimmer light. And a quick note is that frequency and wavelength are commonly used interchangeably, but they're connected, where short waves correspond to high frequency and long wavelengths correspond to low wavelengths. This diagram showing waves makes this clarification clearer and is something to keep in mind. Before learning how transduction takes place, we have to go over some anatomy. So it's a lot of vocab, but the cornea is the clear dome-shaped surface covering the front of the eye. Its job is to refract or bend light to help focus it on the retina. The sclera is the tough white outer layer of the eye, which protects and maintains shape. The iris is the colored part of the eye, and it controls pupil size and regulates how much light enters. And an example of this is when you shine a flashlight, an iris constricts to protect the eye. The pupil is the central opening of the iris, and light enters through it. The ciliary body controls the shape of the lens to focus on far or near objects. The retina, which is really important, is a light-sensitive layer at the back of the eye. It contains rods and cones, which we'll go over later, and it converts light into electrical signals sent to the brain. The fovea is a small central pit in the retina, packed with cones and responsible for sharp central vision. Almost there. The optic nerve is a bundle of fibers that transmit visual signals from the retina to the brain. And the optic disc is the blind spot, and it's the spot where the optic nerve exits the eye. And it's called the blind spot because it contains no photoreceptors. So photoreceptors capture photons of light and convert it into neural activity via phototransduction. The diagram is a bit small, but the zoom in is showing the back of the retina. It works back to front, first through the rods and cones to the bipolar cells and then the ganglion cells. Bipolar cells transfer information from photoreceptors into the retinal ganglion cells. There's also amacrine and horizontal cells, which allow lateral communication between the cells of the retina. Ganglion cells form the optic nerve and send action potentials. So a quick test we can do for the blind spot. This might not work, but you might need to stand up to get it to work. Yeah, it'll give you, you kind of have to move back and forth. And if it doesn't work, you can look this up after because it is really cool. So there are two different photoreceptors in the back of the retina. We have around 4 million cones, which are responsible for photopic vision. There are blue cones, green cones, and red cones that are responsive to wavelengths corresponding to these colors. But it's not just a case of cones being responsible for colors, responsible for color vision, which we'll get to in a moment. We have about 100 million rods, which are responsible for scotopic or nighttime vision. They're highly sensitive but have a low acuity in dim light, lacking detail and color information. So there's about 100 rods to a single ganglion cell versus one cone to each ganglion cells. This is why the rods are so sensitive. The brain can't have detailed images, just the shape when it comes to rods, whereas for cones, you get very detailed color vision. Photoreceptors can lead to a cool afterimage effect, which we'll see in a moment, where cones can get tired and they'll get too much stimulus and stop responding. So we'll see what happens in a moment. So while it's true that we have specializations regarding color, a crucial part of color vision is the acuity and density of cones in the fovea that allows us to see color, since color is a detail. And this graph really shows how there's a huge spike in the fovea of cones. Another thing to keep in mind is that there's a different pathway or a different. Rods and cones are usually the last layer to receive light as it passes through, but for the fovea, it doesn't have to pass through blood vessels or other layers of the cells. So let's do a test. So it's going to be a little awkward, but we're going to look at this for 40 seconds. Look at the pink dot. Are you guys seeing it? Yeah. So you should be seeing an American flag. This is because our red, green, and blue cones adapt or experience fatigue to the colors that you're staring at. So the cones that were being used the most became temporarily less sensitive. For the white part of the surface, while light contains all colors, since some cones are tired, the less stimulated cones respond more strongly. And this is also related to the opponent processing theory of color. Okay, some details about ganglion cells. We have M ganglion cells, which are larger in size and respond more quickly to visual stimuli. Their job is to detect motion and broad changes in images, so they're essential for seeing things like flickering lights or movement across your visual field. M cells receive most of their input from rod cells, which are more sensitive to light but not to color, making M cells ideal for nighttime vision and motion detection. P ganglion cells are smaller and they respond more slowly but with greater detail. P cells help us perceive fine details, color, and texture. They receive input primarily from cone cells, which are sensitive to color and are concentrated in the center of the retina, fovea, supporting high acuity vision. So here is an image of the pathway that how our information from our visual field travels to the occipital lobe. So each eye sees both the left and right visual fields, a part of them. The left visual field hits the right side of each eye, the nasal retina of the left eye, and the temporal side of the right eye. The right visual field hits the left side of each eye, the nasal retina of the right eye, and the temporal retina of the left eye. So to explain those terms, the nasal retina is the inner half of each retina, here and here, and the temporal retina is the outer half, closer to the temples. Here. These regions determine how signal travels to the brain. Then we go to the optic nerve, which carries visual signals from each eye towards the brain. Then we hit the optic chiasma, which is the crossover point. This signals from the nasal retina cross to the opposite side of the brain. Signals from the temporal retina stay on the same side, and this ensures that the left visual field goes to the right brain and vice versa. Then it meets the lateral geniculate nucleus, or the LGN, which is a relay center in the thalamus. This sorts and sends visual information to the correct part of the brain. Then we finally reach the primary visual cortex and the occipital lobe. This is the final stop for visual processing, where the brain turns electrical signals into what we see. And again, this means that your left visual field is processed on the right side of your cortex. So, for the primary visual cortex, we have V1, which is 2 millimeters thick. This is a small region in the occipital lobe. It's the first stop in the cortex. Regions change throughout the pathway. It's organized in cortical columns and retinotopically. So the retina, what's seen on that is corresponding to the V1. There's also cortical magnification, where it goes from simple, complex to end-stop cells, which are most cells stimulated by bars or edges. So I'm sure some connections between computer vision are coming to mind. Both computer vision and our primary visual cortex use feature detection. In this case, the cortex starts off by differentiating large features before focusing on smaller ones. So to go into a little bit more detail, V2 acts as a relay station. It begins by combining features like color, shape, and motion. V3 processes dynamic form, so shapes of moving objects. V4 specializes in color perception and object form. And V5 detects motion, direction, and speed. So we have two pathways called the dorsal and ventral pathways. The dorsal pathway is the where pathway. So dorsal, if you think of a dolphin, their dorsal fin, it's the top or the back of your head. For this, it goes from V1 to V2 to V5 to V6, overall to the parietal lobe. This is responsible for localization of objects in space. The ventral pathway, ventral is underneath. This is V1 to V2 to V4 to the inferior temporal cortex. The ventral pathway is responsible for object identification and recognition. So we're going to go over case studies. So what do you guys think would happen if someone had damage to their dorsal pathway, and where would this damage be? Damage here typically affects spatial awareness and motion perception. So a patient might struggle to locate objects in space, or a person might struggle to locate objects in space or visually guide their movements. For example, they may be able to describe an object but fail to reach for it accurately. This type of damage, actually, did anybody have an idea for where it would be? So remember, dorsal is like the top, like you have to imagine like a fin on a dolphin. Yeah? The occipital lobe is more like back here. So think of something that's one of the lobes that's higher up. Yes. So this kind of damage usually occurs in the parietal lobe. This condition linked to a condition linked to dorsal stream damage is called optic ataxia. So now on to the ventral pathway. What do you guys think would happen if there was damage to this pathway? Okay, so damage to this pathway affects object recognition and form perception. A person might see something clearly but not recognize what it is. Would it also deal with facial recognition, or is that like Yes, it actually does. That is called, it's a long word, it's prosopagnosia, which is called face blindness, is linked to damage in the ventral pathway. So yeah, people might not even recognize objects or faces. So this looks kind of scary, but in this photo, each purple circle represents a receptive field of a retinal ganglion cell, a specific area of the retina that the cell responds to. So that bar of light activates multiple receptive fields, which form the optic nerve, traveling to the LGN in the thalamus, which then organizes the visual system. And in the V1, cortical simple cells receive this input. So you can see here the neuron activities response. It responds a lot when they're straight like this, whereas not as much when it's sideways. So a quick discussion about computer vision versus the visual system. Like CNNs, neurons at different levels respond to more complex features. There's receptive fields involved, and neurons respond to a specific region in the input data. And then convolutional layers use filters or kernels that scan receptive fields across the input data. So a question for you guys. Which of the line parts do you think is the longest? So they are both the same. I think a lot of you have seen this illusion. It's called the Muller-Eier illusion. So those living in Western societies like us in Seattle typically fall to this illusion more often because of the sharp edges within our architecture. So our brains are actually influenced partially by culture in a way that computer vision would not be.